Hello all. Today we are finally going to end 9.1 nervous system, where our final discussion will be on the mechanism of drug action. Examples of drugs that we will discuss are cocaine and curare. For starters, let's see why drugs are bad. Drugs are chemicals that are psychoactive, meaning that they can affect our brain. They affect us firstly by leading to addiction, where we come to a stage where we cannot live without the drugs. The drugs will change our mental activity to become more negative, as well as change our physical activities. So let's try to understand a bit more on what these changes are. We can categorize drugs into a few groups based on their action. Depressants, they will slow down our activity. For example, here you see it can make you fall asleep. This is because the drug can reduce the neurotransmitters released at the synapse. There are many other types of drugs that are depressants, but based on our syllabus, we will be studying about curare, which is more of a poison. Now, the second drug that we are going to study is a stimulant. Stimulant will stimulate our nervous system for us to be active for long periods of time. This is because the drug cocaine, for example, can increase the amount of neurotransmitters at the synapse. Now, there are other groups that are not in our syllabus, but I'll just introduce to you for general knowledge, such as the hallucinogens, where the drug addict will hallucinate. That means they will imagine things that are not there. Then we have a group called the antidepressant, which clearly is the opposite of a depressant, where it will keep you feeling happy all the time. And finally, you have the inhalants. Inhalants are usually the initial stages of addiction, where you enjoy the smell of petroleum-based products like thinners or glue. So now you know the categories of drugs. Let's move on to see how these drugs affect us. Since in our syllabus, we are only going to discuss cocaine and curare, therefore, I will only focus on the stimulants and the depressants. The stimulants and the depressants are effective at the axon and at the synapse, where the stimulants will stimulate transmission of impulse along the axon and transmission of impulse across the synapse. The depressants do the opposite, that is, they will inhibit transmission of impulse along the axon or inhibit the transmission of impulse across the synapse. So let's look at how the drugs do this and how some drugs affect the transmission of impulse at the synapse. There are 5 ways. The first way is by affecting the transport of the synaptic vesicles to the presynaptic membrane. So a stimulant will move the synaptic vesicles quickly. A depressant will move it slowly. Second method is by affecting the release of neurotransmitters via exocytosis. So I'm sure you can guess. A stimulant will release the neurotransmitters quickly. A depressant will release it slowly. The third way is by affecting the sodium ion voltage gated channels located on the postsynaptic membrane. So, a depressant will close the sodium voltage gated channels and prevent influx of sodium, whereas a stimulant will open the sodium ion voltage gated channels so that an influx of sodium occurs and an action potential can be generated. The fourth method is specifically for stimulant, where you see it is able to inhibit enzymes that are involved in the hydrolysis of the neurotransmitters. If you can recollect, at the ending of a synaptic transmission, the neurotransmitters have to be removed. So some neurotransmitters will be digested or hydrolyzed by enzymes in the synaptic cleft, while some of the neurotransmitters will be actively transported back into the synaptic knob to be hydrolyzed by the enzymes inside the synaptic knob. So we see that there are some drugs that will inhibit these enzymes. Therefore, the neurotransmitters will not be hydrolyzed and they will continue to function. So that is why this method is specifically to the stimulant. The final method is the drugs being able to mimic the neurotransmitter. Mimic means copy. So this example here, let's say the solid yellow is the neurotransmitter and this hollow yellow is the drug. So since the drug has a similar shape to the neurotransmitter, therefore the drug can bind to the receptor at the postsynaptic membrane, but the drug will not be able to make the sodium ion voltage gated channels open. They work just like the enzyme substrate concept. So these are the 5 methods in general of how drugs can affect impulse transmission at the synapse. Let's move on to see how drugs affect impulse transmission at the axon. At the axon, the drug can either inhibit the sodium-potassium pumps or they can block the voltage channels, either the gated channels or the non-gated channels. So in this way, what's going to happen is the generation of an action potential is prevented. So most of the time, the drug that disrupts impulse transmission at the axon will be a depressant. Now that we have seen the drug action in general, let's go into the details of each drug according to our syllabus. Start with cocaine. Cocaine is a drug that is extracted from a plant known as Erythroxylon coca. This plant actually is rich in nutrients and alkaloids, but one of its alkaloids is cocaine. Let's have a look at what cocaine can do to us. Cocaine actually has some positive effects. It is a powerful stimulant where the effect of the drug can last from between 15 minutes to 1 hour. So a person who has taken cocaine will experience a lot of positive responses such as feeling very athletic, very energetic. They also will feel euphoric. Euphoric means an extreme feeling of happiness, and they will feel alert. So sometimes people who are professionals have taken cocaine because they find that they are more capable to carry out their duties. But you must remember the stimulating effect only can last about 1 hour. So how does cocaine do this? It can affect the user both at the synapse as well as at the axon. So I'll start the discussion with synapse. Cocaine affects neurons that use dopamine as their neurotransmitter. Neurons with dopamine are found in the brain. So in a normal situation, the brain neurons will release the dopamine for us to have this feeling of happiness. But once the function has been carried out, dopamine will be reuptake into the synaptic knob for use later. It is transported back into the synaptic knob using a dopamine transporter. Now, once you take cocaine, what happens is the cocaine will bind to the dopamine transporter located at the presynaptic membrane. So you see, once cocaine is blocking the transporter, dopamine is unable to return to the synaptic knob. Dopamine will remain in the synaptic cleft and accumulate. So you can see that this dopamine will continuously bind to the receptor, causing the sodium ion voltage gated channels to continuously open, leading to an influx of sodium ions. So the dopamine is going to cause an enhanced and prolonged effect at the postsynaptic membrane, and this will lead to an easy accumulation of EPSP, leading to action potential. So now the person is going to continuously feel happy. So that is how it works as a stimulant. Let's look now at how it works at the axon. When cocaine is at the axon of neurons, cocaine will bind to the sodium ion voltage gated channels. So when it binds to this gated channel, it is going to prevent sodium influx. Without sodium influx, action potential cannot be generated. And this can be good because it can function now as a painkiller. So, a person who has taken cocaine will not feel pain since the impulse of pain is not being transmitted. So, you can see both the explanation I have provided seem to suggest to you that cocaine is good. But let's see what happens when we use cocaine for long term. We look at the negative effects of cocaine. The person will undergo withdrawal symptoms. Example of withdrawal symptoms are anxiety. Anxiety means you feel worried all the time. Tremor and convulsion. This means your hands will keep shaking. Your body will keep trembling. Increased body temperature, so you will be always sweating and feeling hot. Restlessness, you cannot sit quietly in one place. You will always feel your heart racing fast. You'll be nervous. And of course, paranoia. Paranoia means you will be suspicious of everybody. You will be thinking people are staring at you, people are talking about you, people are planning bad things against you. So you will be always living in this feeling of fear when you are not taking the cocaine. Let's try to understand why a person will experience these withdrawal symptoms. This happens when the person is chronically using cocaine. That means you are using a lot of cocaine. You find that the cocaine, once it blocks the dopamine transporter, dopamine will accumulate at the synaptic cleft. Now, the brain will misunderstand this and think that it has produced too much dopamine. So what the brain will do now is its neurons will start to synthesize less dopamine. Okay, that means once the person stops taking cocaine, the person will find it difficult to feel happy because the brain has stopped making natural dopamine. On top of that, the neurons will also reduce the number of dopamine receptors on the postsynaptic membrane. So now the person is facing two problems. One, the brain is producing less dopamine, and two, the brain is not having enough receptors to receive the dopamine. So that is why the person will experience the withdrawal symptoms. And it can be so depressing that what happens with these drug addicts is they will have to start taking higher doses of cocaine. So the more cocaine they use, the more addicted they get, and sometimes the level is so lethal that they end up dying. Okay, so always learn to be happy with the natural amount of dopamine released by your brain. Do not try to use artificial products that seem to make you happy but in the end make you feel more miserable. Okay, let's move on to curare. Curare also is extracted from plants, okay, where they remove a chemical from the bark and stem of plants that are mainly found in the jungles. Okay. Firstly, it was located in South America, but you can also find this in other countries. Now, curare is not a medicine. It was used by the native tribes like the Red Indians or the Aborigines to put into the arrows, okay, that they use for hunting. So in this blowpipe here, there is an arrow, and at the tip of the arrow will be the curare poison. Now, what can this poison do and how is it helpful in hunting? Let's see. Curare also affects the synapse, but curare affects neurons that use acetylcholine as their neurotransmitter. So, the postsynaptic membrane of curare's action is not located on another neuron, but it is that of the muscle. So, curare's action is against a muscle. Curare is known as a competitive antagonist because it is similar in structure to acetylcholine. So, curare here will compete with acetylcholine for the same binding site on the postsynaptic membrane. And you see, curare actually binds faster to the receptor compared to the acetylcholine. This is because the acetylcholine receptor has a higher affinity to curare compared to acetylcholine. Once curare binds to the receptor, curare will block the acetylcholine receptors, thus preventing acetylcholine from binding. So when curare is bound to the receptor, the sodium voltage gated channels cannot open. They will remain closed, and so they will prevent the influx of sodium ions. Without the influx of sodium ions, there is not going to be any EPSP or action potential generated. Without action potential, depolarization of the sarcolemma and the T-tubules will not occur. So, because of that, calcium ions will not be released from the sarcoplasmic reticulum and actin and myosin will not be activated. So, the muscle will be relaxed. Once the muscle is unable to contract, we consider the muscle to be paralyzed. So, curare is also known as a non-depolarizing because it doesn't let the T-tubules depolarize, and muscle relaxant because the muscle relaxes as a consequence of no depolarization. So remember earlier I told you curare is a poison used in hunting. So let's see how this benefits the hunter. The hunter has had a very successful hunt. What has happened is when he shot these animals with the poison arrow, the muscles of the animals became paralyzed, causing them to fall from the tree and unable to run. Then the respiratory muscles also were unable to contract, so these animals cannot inhale or exhale. So that is called asphyxiation, which eventually will lead to the death of the animal. Now, how would we solve a problem if the hunter was accidentally shot with a poison containing curare? Now, because humans are bigger, we would not die immediately like the animal. We can overcome the poisoning. You can use the concept we learned under the topic of enzyme, where to overcome a competitive inhibitor, we have to increase the substrate. So let's think about what is the inhibitor and what is the substrate in this situation. The inhibitor here, of course, is curare, and the substrate is the neurotransmitter acetylcholine. Now, how do we increase the amount of acetylcholine? This can be done by using a protein known as the acetylcholine esterase inhibitor, which I represent here with this orange model. It is actually a protein that functions as an anti-choline esterase. The yellow here is the enzyme choline esterase that can hydrolyze acetylcholine. So, doctors will have to treat this individual by injecting them with a protein that we call the acetylcholine esterase inhibitor. Let me explain how this works. The neuromuscular junction in a normal situation. This is the enzyme and this is the neurotransmitter acetylcholine. So in normal situation, after the acetylcholine has carried out its function, the enzyme will hydrolyze the neurotransmitters, causing their concentration to decrease. But when curare is present, we need to have more of the acetylcholine. So that's why the patient will have to be injected with the acetylcholine esterase inhibitor. The inhibitor will be able to inhibit the enzyme choline esterase so that it is unable to hydrolyze acetylcholine anymore. So acetylcholine now can start to accumulate. The accumulation of the acetylcholine will cause the acetylcholine to have a higher concentration, and that would be sufficient to overcome the effect of curare. Now, these neurotransmitters cannot bind to the receptors that already have curare. However, they can continue to bind to the receptors that have not been blocked by curare. So this way, the sodium ion voltage gated channels can open and we find that the action potential can be generated and impulse can be transmitted to help the muscles contract. So this is how curare poisoning can be overcome. So with that, I end 9.1. Please remember, stay away from drugs now that you understand it better. I will see you next in my video on hormones. Bye-bye.